our next speaker is also from a similar, journey, but a little bit distinct journey. Noah Auerhahn. He is one of the earliest N-of-1 patient parents collaborating with our organization for developing personalized therapy. He is a serious entrepreneur in tech. After his daughter, Ellie, was diagnosed with Rett syndrome, he devoted himself to find treatment for Rett syndrome. He began by identifying repurposed drug that had improved Ellie's phenotype and quality of life. And over the last 2+ years, he has been developing a series of novel compounds that are working in Ellie's cell lines and are now beginning animal studies. He founded Uncommon Therapeutics, a VC-backed startup to pursue his effort to build the curative therapies for Rett syndrome. So,
thank you. Thanks, everyone. First, thanks to a lot of people in this room doing actual work for us. So that's really, really exciting. So started a company called Uncommon Therapeutics to basically kind of house all the work that we're doing. As Yamwei mentioned, basically my entire adult life has been building companies in tech and initially e-commerce and then health tech. Started my first company actually down here in Pacific Beach. So was here from 09 to 15 roughly. And then sold that company to Rakuten and then took some time off, started another company in health tech and tell you about my journey from there. My co-founder, Ryan Lim, is spent about 15 years kind of studying CNS diseases at UCI. And most recently at Module is working on an ALS drug that's going through IND enabling right now. So a bunch of really awesome people behind us and great partners that we've collaborated with over the last almost 4 years now. My background, I was, while I was building my last company, my daughter Ellie was diagnosed with Rett syndrome in 2021. And so I stepped back from my role running that company to focus all my time on building a care plan for her. When you get diagnosed with something like Rett syndrome, which is more common than a lot of the other diseases that folks are talking about, still the general recommendation is PT, OT, and pray for gene therapy is essentially what a doctor will leave you with. And that just didn't really work for me. So Rett's caused by de novo mutation to MECP2. It basically imagine your child is developing normally, and then around 18 months they lose the ability to walk, speak, and use their hands, and then there's essentially a cornucopia of terrible things that happen to them: seizures, scoliosis, dysphagia, on and on. It is a neurodevelopmental disease, so in 2007 there was some exciting work that showed that simply replacing the gene could rescue the phenotype in mice. And so that kicked off a lot of funding. And that was part of my kind of experience going into Rett syndrome, which is, one of the first day or so, Googling or going to PubMed and you search MECP2 or Rett on it, and you'll notice 3,400 papers, 200 new ones every month. At the time, no available treatments. So this is like the dream that everyone would want, their rare disease to get this kind of attention. And yet PTOT and pray for gene therapy was essentially all you could ask for. So I think it's a lesson as we figure out where we prioritize our efforts in terms of what are we bringing to patients. Rett does have its first approved drug. We'll talk about that a little bit later now in 2023. But basically, I dove in deep. I probably read about 300 papers in the first 9 months, met with every expert until I became one. And it seemed obvious to me that we could not develop a gene therapy in a timeline that would be able to slow down this regression, this loss of skills that happens over a period of 3 to 12 months, really. But we could try to bring drugs off the shelf, and so that's what we did. We developed a thesis around oxidative stress, neuroinflammation, a neurochemical imbalance that exists within Rett syndrome, and a lack of growth factors. And so we attacked that with a handful of repurposed drugs in series, and a couple of those medications that have been explored in Rett syndrome in those 3,400 papers, really purely based on lit review, and a couple of which Ellie is the only girl in the world with Rett syndrome currently taking. And so while Ellie is still limited by the disease, here's just a video I took of her the other day walking around our yard. Ellie is 5 years old. She can walk around. She does have an apraxic gait. She has limited hand function, but can touch for yes and no. Limited speech, but things like yes, no, more, done, I do. And you'd be amazed at how helpful those things can be when she can get them out. She doesn't have seizures. She doesn't have dysphagia. She's on her height chart, weight chart, head growth chart. And she's using her eye gaze and is also learning how to read. So her life doesn't quite suck the way that we thought it would, and our life doesn't suck quite the way we thought it did. And that was kind of like the first kind of 18 months was getting to 18 to 24 months was getting to a place of like stability around this. And we're fortunate because it's a neurodevelopmental disease. I think if you're dealing with a neurodegenerative disease, it's a different story in terms of the timelines that you have. But this is giving us an opportunity to then go to the next step of, can we attack the underlying cause of the disease? In this case, the gene of interest, MECP2. So could we leverage, our expertise and the latest technologies to target the underlying cause of the disease? And the more and more time we spent kind of thinking about this, from both a scientific perspective on the disease, but also a business perspective on the disease, on how to attack the problem, is essentially, thankfully the investment community has spent billions building lots of new technologies and ways to build drugs. It's really awesome. And so we have, all these new modalities to attack the problem, yet many of them are not reaching our patients. And then also we have, a lot of really great partners who are building capabilities like here at iXCell, to do specific activities for us that we don't have to build our own lab to do. And so, can we leverage these technologies, tools, and really great people that are available to us to guide us? And so, what we believe is, an alternative perspective is maybe the platform actually was never the technology, it's actually the disease. And in a disease like Rett, every single drug you would bring to market would be worth billions. And the reality is we know that these rare diseases typically require multiple drugs. That's what we're seeing. And so we think it actually makes sense to build a company there. And that is actually where the value creation point is, and likely better for investors because there's just so much dilution that happens here during the platform creation part. And so we've been proving that out. So for about $1 million, we've developed 3 different approaches to addressing the gene of interest directly, and across 3 different modalities that are all working in patient cell lines. So we have an ASO that will increase MECP2. Basically MECP2, the mutation of it, it's a transcriptional factor, kind of epigenetic chromosomal regulator, really important in the brain. And basically the mutation causes it to lose affinity for the DNA, and then it gets downregulated by 70%. In 2017, a researcher out of UPenn showed that simply increasing levels back to normal was sufficient to rescue the phenotype, because it kind of does this, and that's good enough. And that's a much simpler problem in general than trying to replace the gene everywhere. And when you have different communities, it probably took, I think, the Rett syndrome community like 5 years to adopt that into the orthodoxy, where everyone finally agreed on the research. But meanwhile, that seemed like an approach we could take. So we designed an ASO that would bind to the 3' UTR and increase expression of MECP2. And also we did a large screen looking for basically existing repurposed compounds that could also increase MECP2. And then most recently, got some CRISPR base editor data back, working with the folks here, and in collaboration with some folks formerly from Beam to help us design a base editor. And now we have one that's hitting the mutation. Her mutation is the most common Rett causing mutation, which would account for about 10% of Rett syndrome. And it's correcting about 50% in the cell lines. And so all of these we're working to basically take into the animal models of Rett syndrome. Meanwhile, just because we just started at Y Combinator, we're going through their process and have access to like incredible computational talent in there. And so starting to look at ways, if there's ways to use AI to basically predict compounds that could stabilize the protein as well. So one more project to keep us busy. And so why we're approaching this a little bit differently. So one of the things is, I really don't love the word rare. I think it connotes something that will never happen to you. That's why we call ourselves uncommon, because Rett is not rare. It impacts 1 in 10,000 girls. This is the first Rett approved drug. This is their first full year. They've done 350 million in sales. This is the chart that got them approved. This is the chart on a y-axis that is a full y-axis in terms of the impact on the phenotype. So pretty negligible. Meanwhile, 85% of the patients had diarrhea during the trial. So it's questioned whether it's actually was an unblinded trial. Anyways, that's what's available to us. I think what it does show is they at least know how to really run a trial. And there's a strong desire for treatments in this community and the willingness to put up with shit. So I didn't mean to say that, but no, it's and this is not unique to Rett. This is all of us. And so you have a lot of companies. You have Taysha and Neurogene that are working on gene replacement. It's very exciting for our community. We're seeing those go into phase 1. We're seeing early data off of that are very, very small ends. You're seeing some improved hand function. That's encouraging, but you're not going to transform that full chart. We might see a meaningful step, I think, with the gene replacement, but the reality is AAV gene replacement can't get to enough cells with a single intrathecal or ICV injection. It's just not possible. And so we already know that this disease is going to require multiple more modalities until we get to the finish line. So this is the work we're doing, and brought in really excellent people who have done this kind of work before. And basically, now that we have our 3 candidates, we're taking all of them into animal models. So the ASO is in partnership with Loyola. They're bringing that into mice right now. It's already being injected. And the small molecule study, we're doing our first target engagement study right now with Cyogen. And then we're starting AAV manufacturing on CRISPR with the goal of doing talks over the following 18 months and then entering phase 1 soon thereafter, and trying to collect some early patient data and then raising the additional capital that we need. Just quickly, some of the key experimental results. This is our most recent base editor data. Really excited about the over 50% editing and the no bystanders, especially in the context of this mutation. There's only 2 non-A's on either side. So the fact that we could hit that was good luck, but also working through a lot of combinations with the folks here at iXCell. We also designed, these are some of the ASO data that showed increasing levels, 1.5x or greater. And then our small molecule work and even seeing some correction of the phenotype and other downstream genes that we did.